Welcome to our May Native Plant Society meeting. We cut off a little bit of the recording for Zoom, so this portion is a re-recording of the things that we discussed but didn't record. So first, we would like to remind you to save the date for the Fall Native Plant Society Symposium. It is in Salado, Texas, Friday, October 3rd, 4th, and 5th. It is over a weekend this year instead of starting on Thursday. And as that agenda develops, you can find that agenda on the Native Plant Society website. We will also be sharing some of those presentations and more details as they come. I would like to highlight a really great collaboration between Wild Ones, which is similar to the Native Plant Society, is more based in the Midwest. I think it started in Michigan, perhaps, or Minnesota. Wild Ones collaborated with the Native Plant Society and Christy Tenek to create a DFW-specific landscaping plan. It has both hardscape elements as well as the vegetative elements. It has a lot of different areas in the landscaping plan, so even if you don't want to replicate the entire landscaping plan, you can take elements of that, like the pollinator garden area or the shade plant area, and modify that for your home landscape. So there are two QR codes to lead you to either the garden design with the plant list or a webinar talking through some of that design. This is a little plug in preview of our discussion about native grasses with our presenter Eli to plant native grasses. Two really great native grasses that do well in our area are buffalo grass and side oats grama. Buffalo grass needs full sun and only requires about an inch of rain or water a month. It will spread both via stolons and as well as via seed, and is a really great short turf grass and an excellent lawn replacement. Additionally, side oats grama is a lovely grass that is also the state grass of Texas. It is a little bit more shade tolerant, but definitely not full shade, and adds great support to your flowering plants, can serve as green mulch, and makes really excellent part shade borders as well. Now getting to our presenter. Our presenter is Eli Hartung. He was born in Pennsylvania, which is my home state before I adopted Texas. He is a two-time Ann Miller Gonzalez Research Grant recipient, and that's actually how I realized that he was in Texas. Last year, I saw one of the Native Plant Society announcements about the research grant recipients, and I saw his name, and I was very excited. So as of last week, he is now a two-time research grant recipient. So that is really exciting. He will tell you a little bit more about his educational background, but just to highlight, he has his Bachelor's of Science in Botany from Utah Valley University, went on to Kansas State to get his master's, and now he is at Texas State University working on his PhD. So Eli will present on water from stone. Is water and limestone useful to herbaceous plants? And he's going to tell you a little bit about how he got into native plants. That was one of the questions I had asked him, whether he was always into native plants. It was something that just as a kid he was interested in, or there was a moment where he really had that spark. He's going to talk about his greenhouse projects from last year, and then is going to get into his greenhouse projects for this year and how he's tracked some of that water usage. So as I mentioned, Eli is from Butler, Pennsylvania, graduated from Butler High School, the home of the Golden Tornadoes, which during the presentation he mentioned was a little strange to have a mascot that is a tornado. Outside of Butler, Pennsylvania, he hasn't seen a lot of other high school mascots that are a tornado. And at graduation, he knew next to nothing about plants. As a kid, he grew up with a wood burning furnace, so they had to chop a lot of wood, and he was very familiar with chopping wood. But had never really thought about wood or trees when they were alive. And I will now we'll move to the live presentation where he begins to tell you about his journey into native plants and how chopping wood led him to where he is today, studying native Texas grasses.
No worries. But I was and kind of wanted to dive into what botany had for me and what other things I could learn in the field. And so about that time, I think the following summer is when this happened. I started working with and volunteering with the Utah Native Plant Society and one of their native gardens that they had at this elementary school. And so I got to learn a lot about, penstemons and eriogonums and astragalus and a lot of cute little Utah plants. And I spent a lot of time here. I spent most of my undergraduate volunteering here. It was with elementary students. We brought elementary students out into the garden and taught them about native plants, which kind of typically translates to keeping them from pruning themselves and pruning each other. But it was a very good experience, and I learned a lot about native plants there. And so this connection, my boss at the Utah Native Plant Society connected me with the Forest Service and Dr. Susan Meyer. And so fall 2018, I started working with the Forest Service in this Forest Service lab with Dr. Susan Meyer, and she ran an endangered and rare plant lab. And so she focused on a lot of strange and rare and threatened plants in Utah. And so we worked specifically with 2 astragalus species, a Phacelia, and then this plant Arctomicon humulus, the dwarf bear poppy. And this plant I really fell in love with and ended up kind of picking a project on during my time with the Forest Service. And so this plant, if any of you have been to St. George, you're in the heart of its range. It grows in one county in Utah. And it grows on these kind of gypsum outcrops in the Mojave Desert. So it's highly restricted to these very kind of weird, intense soils. And I was interested in how their seedlings establish and things that the seedlings like, basically. And you can see in this picture, there's a penny next to one. These little guys were very hard to find in the field. But it was a very fun project. I learned a lot about rare plants. And this was a really important experience because this was kind of, as I was learning about botany, I was learning about it kind of through a rare plant endemic lens. And so also during this time, during my undergrad, I had an introduction to grasses. This was a course taught at my undergrad that I took. And I really didn't know what this course was about walking into it, which I think was a good thing. I think if I had known to be afraid of grasses, I wouldn't have taken the course. But I took the course and I really liked the course. Similar to the wood thing, I had never thought about grasses beyond my lawn. And I learned about all the kind of differences in form and diversity in the family. And I really fell in love with the group. And so from that class on, I had the kind of focus of, I want to do my master's with grasses and learn about wild grasses specifically. And so I wrapped up my undergrad in 2021. And then I moved to Kansas. I really wanted to be in the American grasslands. And so I went to Kansas State for my master's in 2021. And this is probably the part of the presentation where you start to recognize more of the plants than before. But really getting to know some of these really cool, really kind of robust prairie plants that are like the hallmark of the American grasslands. So it was a great experience there. And my specific project was looking at how Andropogon gerardii, or big blue stem, responds to soil communities and soil microbial communities. And if you've been up to Kansas, you know it kind of has the same rainfall gradient that we have here in Texas. And so we were interested in if plants in kind of the eastern portion of Big Blue's gradient like their soil communities compared to like a western community. And so this was also a cool experience. I grew a lot of Big Blue stem plants. And so wrapping up this project, I wanted to keep going with grasses. I really liked my master's project, and wrapping up for my PhD, I wanted to keep going with them. And so about the time I finished up my master's, I got in touch with Dr. Susan Schwinning at Texas State, and that's where I'm at today in their PhD program. And the first things I noticed here, I hadn't been to Texas before I came to visit. And the first thing I noticed when I came to San Marcos was the limestone. I had never been in a limestone dominated system like this. And Utah has rocks, but not like this. So this was really new to me. And it was also new to me of just the diversity that we have on the Edwards Plateau and all these really cool, rare plants that aren't found anywhere else in the world. And so as I'm coming into Texas and I was learning and thinking about my projects, a lot of these old ideas that I had from Utah and Kansas started to kind of come back to me. My lab specifically focuses on plant-water relations and below-ground processes, but Texas and the Edwards Plateau is kind of this cool mix of the northern prairies coming down, getting those tall grass prairie species, but also a lot of things that reminded me of Utah. We have those cryptobiotic crusts here, and a lot of species that reminded me of Utah. And just kind of looking at a limestone outcrop here, this felt a lot like an endemic system to me. Low plant density, high exposure. This just kind of was like yelling at me like endemism questions are here. And so what I wanted to do for my PhD was kind of combine plant water use in grasses with like an endemic plant lens to see what that could tell me about plants here. And that's basically where these big questions that I had for my dissertation come of, can water and limestone benefit grasses during drought? Do plants on the Edwards Plateau have adaptations to extracting that water? And can that limestone and that water dynamic shape the diversity we see on the Edwards Plateau is kind of the overarching questions that I want my dissertation to kind of poke at. And so that's where we are. And so that's kind of about me. That's how I got here. I'm going to take a sip of water, and then I'm going to talk about some of the projects themselves. And so as we get into my first research project that I want to talk about, I just want to go over a couple topics and concepts. So you're probably all aware of plant available water. But plant available water is basically just water that plants can use. So if you had like a pot, when you put too much water in, it flows out to the bottom. That's kind of above field capacity, so plants can't use any water that flows past their root systems. But there's also kind of a lower bound on that where if the soil's too dry, the plants can't extract it away from the soil. And so this is typically how we think about plant water use. We define it in terms of plant available water. But there's this additional issue of accessibility, which is kind of obvious in the Edwards Plateau, of whether or not a water kind of volume is extractable by plants doesn't matter if their roots can't get to them if it's stored in limestone. And so this is kind of something my lab and I think about a lot of. This doesn't matter as much if the roots can't get there. And this translates, and a lot of people have thought about this because it applies in other systems where, I think I broke my pointer.
There was you clicked something and then the screen popped up. Let's see, there we go.
Sorry about that. Anyway, but so this idea of accessible water and available water has kind of been played around with some other groups. And there's a group led by a guy named Rael in Utah who thought about this, where we can think about accessible water and available water in terms of kind of pools, where growth pools are typically in upper soil layers. They're really easy for plants to access, so they use them while they're growing. And we can think about this as upper soils during the growing season. Rainfall comes into those upper soil layers, plants take them up immediately, use them to put out new leaves, flowers, fruits, etc. And we can compare this to something like a maintenance pool, which is harder for plants to access. And because of that, it sticks around longer, like a deep soil or potentially a rock. And there's kind of empirical evidence to support that this works. plants switch to deeper soils as kind of the upper layers dry out. And on the Edwards Plateau, there's evidence that trees here switch from soil during kind of ideal conditions, and they'll pull some water out of the bedrock during drought. But the issue that I want to pose, and the kind of issue of my dissertation, is this conceptual diagram that talks about these pools and whether or not they exist just totally ignores grasses. It just assumes that they don't make it into maintenance pools, that this concept kind of doesn't apply to them at all. And that's what I want to think about for my first project, is potentially limestone or water in limestone, which depletes at a slower rate than the soil, could function as a maintenance pool for grasses here on the Edwards Plateau. And that's kind of the big questions that I want to ask is, does this apply to herbaceous plants? So we see that in live oaks. Does it apply to grasses? And so this is where we get into the summer 24 greenhouse projects. And so for this, I want to talk about 5 experiments that I conducted last summer, and then kind of synthesize them all together. And again, thank you to the Native Plant Society for funding this project. But so basically the idea for this project last summer was, I wanted to ask the question, does limestone, or do limestone and endemic grasses benefit from limestone in soil? Was basically the main question, and if so, how? I was kind of walking into this blind, and so I wanted to see what I could find out that way. And my main hypothesis was that endemic growth would be better than just based on total available water. And so in like kind of a hypothetical results section, I expected that some plant response under well-watered conditions wouldn't differ between plants growing on limestone or soil, but in drought conditions, these endemic plants would prefer growing on limestone for some reason. And what that is, I wasn't sure at the moment. I break this again. Okay. Perfect. And so the idea was this was going to be a greenhouse experiment, so I could manipulate these pots pretty well. And so what I wanted to do was, from my field site where I collected seed, mimic the soil rock fractions. And so this is kind of what that middle picture is representing. So my field sites had 30% rock or limestone rock in those upper soil layers. So I wanted to mimic that in pot. And then I additionally found that there was 150 milliliters of water stored in both the soil and the rock combined. And then I wanted to compare that to a full volume of soil, which held 190 milliliters of water. And then I wanted to compare that to a similar rock fraction, but with granite, which it doesn't hold water. And so this design would help me kind of identify rock presence absence, as well as rock water presence absence, and how those things affect these plants and how they grow. And so I wanted to work with a grass, obviously. So I chose Muhlenbergia repertonii, or seat muley. And I chose this plant mostly because of its range. Kind of the core of its range is along the Edwards Plateau, going up into the limestone grasslands of Oklahoma. This seemed like a candidate to tell me something about what plants like about limestone. And so I picked this plant. And how I'm going to do this is 4 of the 5 experiments are kind of in pairs, so a 2-2 kind of design. And so what I wanted to do for the first 2 experiments was understand how these plants function under well-watered conditions. And then kind of the results of the first experiment would design the second one, or like inform the design of the second one. But so the first experiment was just grow muley plants in these 3 treatments. And if I found a limestone effect, I would crush the rock. There's a confounding effect of limestone could be good for water, but it could also be good for nutrition. But crushing the rock would delete that water effect and just keep the nutritive effect. And so the combination of these 2 experiments should tell us if limestone helps plants grow here and the cause of that effect, if there is one. And so here I have the first experiment. I have total biomass on the top and root shoot ratio on the bottom. For the full soil in red, the granite in white, and the limestone in blue. And so for total biomass, we see full soil plants are the largest, the granite plants are the smallest. And this kind of makes sense. This matches our total water capacity of our pots. And this made sense to me when I weighed them and kind of found these results. But what was interesting is the root shoot ratios and where this biomass was allocated differed between the treatments, where the full soil and granite plants have about a 1-to-1 ratio, but the limestone plants heavily produced below ground. They were producing much more roots than the other two treatments. And this is interesting because this is common in drought systems and in arid systems as like a drought effect. A lot of desert plants heavily produce below ground. So it was interesting to see that here. And so we see that limestone effect in our first experiment. We moved on to our second experiment where we crushed the rocks. And what we found is when I crush rocks, I lose those effects. Total biomass isn't different between the three treatments, and root shoot ratios aren't different between the three treatments. And so the combination of these two experiments tell me at least that limestone is important for the muley and how it grows, and something about the structure of that limestone is important. And so taking that and kind of keeping that in our back pocket, the obvious question is, we see this in drought adapted plants. How does drought affect these kind of interactions? And so I did another 2 experiments where I did those same 3 treatments and just wanted to see how long they could survive in drought. And then again, if I saw that limestone effect, I would look closer at the physiology of these plants during that time. So I wanted to do some photosynthetic measurements, gas exchange. But I'll point out that I included a 4th treatment in our 4th experiment where I reduced the soil volume to have the same water capacity as the limestone. So it was a full volume of soil, but it held 150 milliliters like the limestone treatment to try and help get at that idea of kind of pools where in our reduced soil, all that water is stored in a growth pool versus in the limestone treatment, it's split between 2. And that was kind of my idea in designing this experiment. But for the 3rd experiment, I found that survival time was greatest in our limestone plants. And so here is survival and then time in days. And so for the blue line, which is limestone, we can see that limestone plants live longer than the other 2 treatments. And interestingly, they outlive the granites by at least 4 days. So it's kind of this really interesting, really strong response. And this is interesting because this doesn't match our water holding capacity in our pots. These limestone plants don't have the most water, but they are living longer than the other 2 treatment groups. So that was interesting. We saw that limestone effect. We moved on to looking at some of the photosynthesis data. And so this graph kind of has a lot going on in it, but hopefully I can explain it well. But so I have photosynthetic rate on the y-axis. And what I wanted to do was take gas exchange measurements during both well-watered and droughted conditions. And so I took photosynthetic measurements, droughted the plants, brought them back up, droughted them again, trying to mimic some of the kind of variation in rainfall we have here, where we get a big rainfall event, it dries out, rains again, goes again. And so I wanted to kind of mimic that in my pots. And so that's what I did. And so the 2 red squares represent those drought treatments. And what we found here was during well-watered conditions, our 4 treatments didn't differ, which I interpret as when water's plentiful, everything's good, plants kind of do well. But what's interesting is during drought, the limestone, which is blue, really does well compared to the other treatments and kind of sits on top of them throughout the droughts. And so plants on limestone are able to maintain higher photosynthetic rates during drought than the other 3 treatments we had. And more interesting than that is, here's that reduced soil treatment, which has the same water holding capacity in black. And you can see that this treatment absolutely tanks during drought. And so this is really interesting because these 2 pots hold the same amount of water, but they're functioning totally differently during drought. And so my takeaway from this, or at least for me, was that the water being in the limestone is important to how these plants respond to a drought. And I just wanted to show you what those root shoot ratios and biomass looks like for those 2 experiments. And so what we see for experiment 3 and experiment 4 is that after drought, biomass is not different between any of those treatments. And I think this is a kind of compensatory effect of limestone, where plants on limestone enter drought smaller, but they maintain higher photosynthesis during drought, and by the end, they're larger. And so kind of at all said and done, growing on limestone isn't too bad for them. But we do see, again, really strong allocation differences, and those limestone plants are producing a ton of roots relative to their above ground tissues. And so that was interesting to see again. And so I just wanted to show you a couple of pictures of what this looks like. These limestone plants were very cool, but a little bit of a pain to harvest because of things like this. They had this really interesting morphology where their roots would kind of wrap around and grow into these limestone rocks when I was harvesting them, or before I harvested them, and then getting them out was a little bit of a challenge. And just moving into my conclusions for the muley stuff, here I have a limestone plant and a full soil plant, and you can see the magnitude of difference in root production between those two plants, which is pretty kind of incredible. But just to conclude on the muley stuff before we move on, limestone plants grew slower before drought, but they kind of had but they had the same biomass after the fact. And then additionally, water being in limestone seems to be important for these plants, specifically for that photosynthesis data where the two pots that had the same amount of water totally responded differently. And so moving on from this, the question I asked next was, what makes these roots grow? If you remember the second experiment where I crushed those rocks, we didn't get any kind of increase in root growth. So something about limestone increases root production, but if you crush it, you lose it. And then I was also interested in other traits, and then I was interested in other species. And here are just a couple more of those pictures of roots growing around limestone. And so this is where I get into my fifth experiment, where I wanted to look at other species. Take another. And so the last experiment I did last summer was looking at other species and if other grasses could do this, and other grasses responded to limestone like the muley does. And so my idea for this was that karst plants would like limestone, prairie plants wouldn't. And so I designed an increased, I'll get to this. Actually, I'm going to go to the species and I'll come back to the hypotheses. But I included 3 more species in this study. So I used the muley again, it's been working really well for me before. And I wanted to include 3 other plants to look at and see how they responded to limestone. So I included Sargassum or Indian grass as kind of my prairie species, silver blue as kind of a generalist, grows on the plateau, grows in the prairies. And then I also wanted to include KR as this. I had this grand idea that if I could show that KR was using our native plants water use strategy, that could help explain why it's so invasive here. So I wanted to use these 3 species in addition to the muley as kind of a gradient of likes karst to doesn't, how do they respond to limestone was kind of my thinking. And then for my hypotheses was, again, well-watered conditions, everyone's happy, but during drought, the plants will kind of flip, where karst plants like the limestone and prairie plants like the soil. And so what I did for this was those 4 species. I just used the 3 rock treatments to keep our number of plants down. And we had 120 total plants. And then the design was similar to our 4th experiment, where we had an establishment, well-watered period, drought, recovery, drought again. So trying to, again, mimic kind of rainfall variation here. And I don't know if you can see it well, but here's a little muley plant. And then everything that's poking up top are all those Indian grass plants that were gigantic by the time I took this picture. But here I have just the results for that. So here's total biomass. It's a little bit messy, but as you can see, KR and Indian grass are bigger than muley. That was pretty interesting to see. But what we do see is that typically across the species, within a species, there's not an effect of limestone on total biomass production, which was kind of interesting. The Indian grass didn't like the granite, but the biomass data in general is a bit messy, and I haven't exactly figured out the story behind it. But what is interesting is the root shoot data. And so we again see in the seat muley that heavy production in below ground biomass, and we see to a lesser extent silver blue stem doing the same thing. And so these plants were collected from the plateau, and so it's interesting to see kind of to a lesser extent a generalist doing something that the muley's doing. And though not significant, we do see in the other 2 species a little bit of an increase in biomass production on limestone. So there might be in general an affinity or some kind of cause of limestone that increases plants' root production, and whether or not plants respond to it, I'm not sure. And here's photosynthetic data. This is a little bit hard to see, but it's structured the same as my 4th experiment. And so it's just split by species. So here's the muley again in well-watered and drought conditions. We keep that same well watered, everyone's the same, and the limestone plants do really well during drought. And then if we compare that to Indian grass, the opposite is true. So those full soil plants really do well during drought, and we see kind of that opposite effect, kind of supporting the hypothesis that we had going into this. For the two Bothria cloas, they're messy and intermediate, and there's not an obvious pattern here, but I think that makes sense for both of them. Silver bluestem's a generalist, not really specialized to either system, and so it just kind of goes as it can. And KR is invasive, and I don't understand it. So it is just, it doesn't cooperate at any level. And so this is what we found from our fifth experiment. And then here's just I think this is a silver bluestem in its root system. And I don't know if you can see it, but there's more fine roots when it's grown on limestone than on the granite. Did I break it? The laser pointer works. Okay, this works. And then, yeah, just kind of the last big takeaway from that experiment was that the gas exchange patterns were different between the muley and the Indian grass, which I think makes sense.
Indian grass collected on the plateau, or was that blackland?
It was collected in Blackland Prairie. Sorry, I did not mention that.
Was it from seed or?
They were all grown from seed, yes. Sorry, I should have mentioned that. So moving on from this experiment, my remaining questions were kind of getting at timing. I didn't have really good data on when these processes take place. And so I was interested in when limestone starts to be helpful for these plants. Is it helpful in well-watered conditions? Is it only helpful in helping them stay alive during drought? I'm interested in other plateau grasses and other plants that are kind of endemic to the plateau. And then I'm interested in getting more specific traits and looking at other things that could potentially be adaptive to plants on the plateau. And so this is where I get into my next steps and kind of what's going on this summer. And so the big experiment for this summer is the greenhouse experiment. And so kind of with my remaining questions, the goals of this experiment are to get better time series data, get better species comparisons, and look at root traits more closely. And so I personally think it's a little bit strange that I compared Indian grass and seat muley, and comparing them is a little bit hard. And so what I wanted to do for this summer is, I'm going to do two things, is I want to do a better job of comparing karstic plants with prairie plants and compare kind of more phylogenetically similar karst plants. And so I want to compare seat muley with Gulf muley. I want to compare hairy grandma, I think is hirsuta, with, I'm sorry, I'm very bad with common names for some of these. Is it blue? Okay, blue grandma or Butylogracillus. I'm just going to go to scientific names, sorry. Carex penistachys with Carex tetrasachia, and then karst. Now silver. I'm sorry. Now I'm flipping back and forth. Silver bluestem on the karst, silver bluestem on the prairie. And so the idea from this species comparisons and this set of species comparison is I can look at multiple karst species or karst populations and compare them to something that's similar to them to see if there's kind of a direction of traits where, you know, Muhlenbergia in general looks like this. To get on the plateau, it's done this. And then I can also look at other karstic species to see if that rooting response is kind of a generalizable trait for most karstic graminoids. And so this was kind of the design for the species-wise, and then specifically looking at silver bluestem more closely as kind of like an ecotype comparison where populations on the plateau might be better at it than populations that aren't on the plateau. And then back to that time series data, I'm going to plan on doing sequential harvests so we can get a better idea of when these kind of root patterns develop and when these differences in biomass allocations happen, maintaining that similar kind of variability in rainfall. And then pot design is going to be similar to last summer where we keep that limestone treatment. But what we're going to do this summer is we're going to combine a couple of those treatments as well. And so we have that granite rock treatment, but we're going to make the pot size bigger so that water holding capacity is the same between the two. And so we're kind of collapsing rock presence absence with total water capacity just to save a couple pots this summer. And the good news is this experiment's already started. So these are going, the grasses are very happy right now and they're all growing. And Emily and I have talked about this, but if you're a sedge person, please talk to me because the sedges are, they're very tough for me to get to grow. And I feel like all the literature on them is about northern sedges. And so the sedges that we have here, just nobody knows, which is all the more reason for us to grow them. But yeah.
I texted him. I'm waiting to hear back.
We're waiting. So the sedges are waiting, but the grass is all growing. And it's actually kind of nice that the sedges didn't go because I'm running out of space on this table. So more on that soon, but this is where we're at on the greenhouse experiment. And then I just wanted to quickly talk about some of the other greenhouse experiments. So the undergrad in my lab is working on a couple experiments this summer as well. So he has two questions. He wants to get closer to what causes that rooting response. So what he's doing is he has all these acrylic cubes and he's just using different grit sandpaper on them and seeing if he can elicit with one specific or a couple specific sandpaper grits that rooting response we saw before. And if there's a specific roughness that these plants are responding to. And he's also doing a second experiment where he's growing these plants across two pots, where in something like this, where it's half in full soil and half in a limestone soil, does a single plant differentially root depending on what it's growing in? And so kind of a scaling question he's trying to get at this summer. We're also we're trying to track some water usage and get a little bit closer to pointing that it is water in the limestone, and that's the driving effect. And so this is through the Texas Ecolab program. I'm working with 7 properties in Burnett, Blanco, Hayes, and Travis County.
What the Texas Ecolab program is? No.
So the Texas Ecolab program is basically, it's a program for landowners that if they enroll, they can get, I think, tax credits, and they can invite researchers out to their property to do plant or animal or any research that they're available for. And so researchers submit proposals, and landowners pick which ones they'd like to have come out.
University, or is it like
It's statewide.
any university, I guess, could tap into it?
Yes, as far as I know.
University specific, it's just a Texas program.
Yes, it's definitely not specific to Texas State. It's through, I think, Braun and Gresham are the attorney's office that manages it. I can share the website with anyone. But it's a cool program, and it gets me out, and it gets some landowners who are interested in plants kind of involving their property with the program.
So if your land loses their ag valuation, this can get you closer back to zero. This is cool.
We are beyond.
So
I know that this gets me out there.
what do you have? It's Texas Ecolab program? I had never heard of it. I feel like this should be on every website. It's awesome.
They're making a new website, so hopefully it's better than their old one, because their old one was a pain. I mean, I knew about the program and I couldn't find it. But it's cool, and if you have a property and you'd like research on it, I recommend looking into it. It could be kind of what you're looking for. But this is kind of the project in general is we're going to use some water chemistry, and the idea is that you can match the chemistry of water in a plant to different soil and rock layers. And so that's what we're working on this summer is sampling plants and rocks and soil under well-watered conditions, and then progressively into a drought to see if there's like a switching effect where when soil water is available, they use it, and then they switch to rocks when it's not. But that's basically one of the ideas that we're working on this summer, and sampling is going on. And then the last one is this field study that I'm working on where I'm trying to get better at comparing herbaceous plants to woody plants. And so basically the idea is we're monitoring herbaceous plants, we're monitoring woody plants, how they're using water and putting or transpiring. And then we got some funding from the university to drill these 24-foot holes so we can monitor how deep the water goes into the rock. And so we're trying to link basically how deep a rainfall goes to what it kind of activates in the plant community, and kind of pulling those three things together. And so that's ongoing right now. And so I just wanted to wrap up by kind of putting it all together. So my dissertation, kind of in a nutshell, what I want is to increase our understanding of wild grasses and forbs. I feel like, especially when we compare them to trees and crop plants, especially, a lot of these plants we just don't know anything about. And I kind of have a chip on my shoulder to kind of defend our native plants and learn about our native plants. And I really want to keep learning about seat muley. I really like this plant, and I think it has a lot to tell us, and so I'm going to keep working with it throughout my dissertation. And then pulling back in these endemism concepts into water dynamics. And so typically when we think about endemism, we think about, soil chemistry and, nickel-rich soils or something weird in the soil. But I think this is a likely candidate for why we see a lot of the diversity here. And I want to keep looking into that. And so with that, I broke it one more time, but I'm almost done, so I won't break it again. Yes, actually, it's just 2 slides. Can you just click? So I just have a couple of acknowledgements. My lab, the Schwinning Lab, and then I also need to thank Texas State, the Texas Academy of Science, and the Geological Society for funding this work, and especially the Native Plant Society. They funded both of my research or greenhouse projects. So thank you all for being part of the Native Plant Society, and thank the Native Plant Society of Texas for supporting my greenhouse projects. And so with that, if you have any questions, comments, ideas, I'm happy to hear them. I love ideas, so just let me know, or you can email me. This is my email. So, and that's all I got for you today.
We have some questions in the chat, and I feel like, so the first question is, have you quantified the water uptake of the limestone?
Like how much water's in it? Yeah, so those pots have, from last summer's greenhouse experiment and this summer, the field site's limestone contains about 11% by volume. And that's how we calculated the 130, the 150 total in the pot. But so about 11% by volume is what those rocks hold.
So if we are doing plant rescues at limestone sites, would it theoretically make sense to take some of those limestone rocks and it would aid in recovery when they're essentially trying to regrow their roots?
I think it is one of those, how much limestone rock do you want to lug?
30%?
Well, I just like, I mean like if you're restoring like an acre and you need to bring 30% of an acre of limestone rock with you, I think it can help. I think whether your back can handle moving all that limestone rock is the question.
Plant rescue will often dig up like 10 or 15 plants per person.
Mhm.
So we can stick it in the pot and wheel it back or whatever.
Yeah, I think I think maintaining something similar to where they're from is the best. And like there's the like local seeds for restoration kind of idea. I think it's kind of goes the opposite way as well. It's like kind of trying to maintain the physical properties of where they were and keep that as well, if possible.
Couldn't you use expanded shale in that situation? You're transplanting plants. I mean, expanded shale is a
But I think
product of concrete manufacturer, which is not.
Water, expanded shale, limestone.
Probably less.
Yeah.
in one of your experiments, you shattered the limestone. It was a bunch of tiny little limestone pieces and the effect was lost, right?
Right.
Oh, were they, was it like?
Less than 2, well, like less than 2 millimeters. I put him through, I put him through a sieve.
Did you find that the difference in root size between limestone and prairie soil was true for both karstic and prairie plants?
I felt, I don't have like the data support this, but I felt like in general, plants on limestone made more fine roots. That seemed to be in general kind of the rule, regardless if they were from prairie or karst. And then in general, the karst plants made more roots, but I felt like as I was harvesting them, even prairie plants were producing more fine roots.
Okay, and my follow-up question is that increased root mass on limestone due to the roots having to go further to access the water?
I think that's a good question. So the pots were kind of built in, I call it my parfait design, where it was a layer of rock, layer of soil, layer of rock, layer of soil. So I don't think that would be why. It could be, but I tried to design so that it wasn't just they're sitting on a big rock and all the soil that's holding the water is at the bottom. That's what I tried to build the pots away from.
I was always also thinking of the structure of rock versus the structure of soil, and thinking that the pockets of water in rock would be further apart, and so the roots would have to go further, maybe.
Yeah, and I think that might be, I think there's the idea that like a root kind of grows through the soil at like a normal rate, but if it hits something, it's like this isn't the place to go, and it kind of increases growth in a different direction. I think that is also part of it, but we didn't see that in our granite rocks. We didn't see more root production. So maybe somewhere else, but I don't think for our plants. Is there a question in the back?
I spent an entire summer when I was an undergrad scanning in the roots of seedlings, and I wondered if it was similar.
So that's why I don't have anything beyond the biomass for it. I tried to scan them and I broke the scanner and like kind of destroyed like a lot of the roots. So I know how to use the scanner now. And so that's one of the things I want to do this summer, but it was just biomass for the roots last year.
The moisture content that you found, when was it taken, and did you do a comparison of before and after the start and the drought condition?
So
to prove the moisture content of the soil versus what was in the limestone.
So what we did was to initially determine the water content. Well, we wanted to determine the maximal water content. So I saturated them under vacuum. We put them in a big jar and sealed it, and then vacuumed out all the air to fill them. And those were the rocks we used at the start. So the rocks started out saturated, and then I did additional experiments to show that if they were, I recovered them mid-drought by kind of setting the pots in water, like capillary watering. And that in a 24-hour period also saturates the rock. So at the start of the droughts, they were saturated. And then I had an issue with the masses. I tried to mass them through the drought, but I think what happened is I lost some rocks. I don't know where they went. I think they fell out of the pot. I tried to get the masses, but it didn't work very well. So I'm trying to be more careful this year about keeping the rocks. Does that answer your question?
Trying to compare so you could try to extrapolate evaporation rate of the soil versus the limestone, and that's why they went to the limestone, because there was more moisture content.
Oh,
during drought conditions.
I see. So that's more what the EcoLab project is trying to get at, is we are tracking the water content as well as the plant water. And so that's more that project where when we're sampling, we're keeping track of how much is in it at the time of sampling, so that we can connect where the plant sourcing as well as how much water is in each limestone or soil. So I don't have data on that yet, but I'm hoping to have that soon, in a year.
A lot of interesting findings from the experiments, but I was wondering, how do we apply the findings?
Good question. I think the application is that Actually, I don't know what most landowners do here, but like in Kansas, in the Flint Hills, everyone removes their limestone from the soil. And so if I was in Kansas, I'd probably say you don't have to remove your limestone from your soil.
Because they think it's bad?
Mhm. Yeah. If they're gonna do anything with it, but that's besides what, it's very hard to do, so most people give up on it. But I think an application of this is that we're learning more about grasses, but also that that like where water is stored can be important, and that, and I was talking to Emily about this with my brother of like what he's planting in his yard up in Austin, or down in Austin at this point. He can kind of maximize that based off like whether or not he picks natives or kind of ornamentals. Is, I think gardening here, where a lot of people are on the plateau and trying to plant, water heavy using plants, they could, I think this is a potentially selling point of like native plants is your native plant doesn't need any kind of like soil amendments to grow here. I guess you already knew that before I started talking, but I'm not sure about kind of big scale applications. I kind of focus little, so I haven't really thought big picture beyond that.
It's a really good point to make, that you should focus on, local ecotype plants, because you know that they're gonna thrive and succeed. So that's another thing. Yeah, great.
Well, that Austin and the Edwards Plateau people don't have to pickaxe too hard.
Right. Well, yeah. Well, he's trying to put rose bushes in, and he's sad that his soil, so he's like bringing in soil. I'm like, you don't have to do any of this. You could avoid all of this. I mean, so yeah.
What kind of water did you use? Was it rainwater or, city water or distilled water or what?
So our greenhouse is off a well, so it was well water. And I haven't looked at any other water, but at least the greenhouse plants. So I haven't used any other water. It was all that.
These are these wells that tap the water from the plants you were experiencing.
I have absolutely no idea.
Well,
I'm not sure. Good question. Yeah, so the well, yes, the well is beneath the university. The university has a couple wells.
Have you looked at, so you're looking at the state muley, which is very like habitat specific. Have you tried looking at other species that are, I guess, more gregarious, but like are more diversely distributed and can thrive on different types? So like the silver bluestem. I don't know, would you try picking plants that can do that same thing and see how they react to the different treatments?
Yeah, so that was kind of the idea for the silver bluestem inclusion this summer, is because it's a little bit more generalist, and comparing those kind of populations ecotypes, I think could get closer to that. But other than the species data that I showed, I haven't included any other, I haven't grown anything. But if you have suggestions, if any of you have suggestions, I love putting plants in pots. So I'm happy to, if you have any ideas for plants, I'd love to know.
You trying to make a statement for all limestones in Texas?
I don't know about other limestones. I know a little bit about Edwards limestone. I'm not sure about yours, but I imagine the muley, since its range kind of extends up through here, it's probably doing a similar thing along its range, but I don't know.
A whole lot of difference in the mineralogy and the porosity.
Yeah, and so I definitely can't speak for the ones that come up here, but that might be a good question, is how do these, like, especially this muley, which range follows a lot of different limestones, is how does it respond to different ones?
You see the map, which ones go with what limestone, like the Kiamichi or the Edwards or whatever. They're all different, and they're 150 million years different in limestone. Therefore, the mineralogy is quite different.
Gonna write down something real quick. Giving me an idea.
Or how one ecotype does with a limestone that it does not normally grow with, and does it have the same benefit or do they have to match?
So all the different sites where the seat muley is found, are all those on the same type of limestone or are they on different limestone?
So everything I collected is from one's population. So I don't know about other seeps, but that's also a good question. It's different. This is very helpful for me too. You also have you guys are also asking me very hard questions, which I appreciate. Are there any other questions? Again, I appreciate you all having me. This has been very helpful, and I'm always excited to talk to plant people.
How do you feel about that grass photo and their roots of native grasses?
Oh, the prairie ones?
Yeah.
Well, so this is going to be like, this is,
As long as all the variability in the link with stems.
This one.
The roots.
Oh, geez. This one.
so this, I'm not going to give my opinion. I'm going to give my plant ecology professor at Kansas State's opinion. He had an issue with these figures because a lot of the times they grow these plants in tubes and that's how they get like maximal depth. And that can be misleading because a lot of these plants heavily root laterally. And so while this is definitely possible for these plants, the most kind of efficient strategy is to grow outward and catch most of the incoming rain. And so Big Blue, for example, typically is much more laterally rooting than deep.
That's more of a hypothetical illustration of root length rather than root distribution.
Right, because typically, like, I think you can imagine for most of these, it should be kind of like a semicircle, and they're kind of a three-dimensional sphere, with most of it being in the upper soil layers, trying to catch the next rainfall event.
But what about the guy that's standing next to the bearded guy that's holding the thing? Those aren't in tubes. They're, twice as tall as him.
What about biomass as well?
Yeah, I don't have an issue. Jesse Nippert at Kansas State had an issue with these. Send your complaints to him.
Brother who's case.
I think it's a great illustration of the variability of roots.
I like it personally. I have a poster of that. The root.
Rather than contrasting what Bermuda grass roots look like with blue stem grass, it's just being controversial.
Can go this deep.
There's a little asterisk. Fix it.
Actually look not through the rock, but to actually figure out how deep the roots are going into the rock at the sites.
So if I was incredibly rich, if I had unlimited money, I could, because there's like, you can like do like wave technologies to like kind of pulse through and get like changes in substrate with depth. But to get like finer scale, you need better machinery and that costs more money. But I can get very low resolution, which doesn't tell you anything about roots because they're thin. So I would like to, but
It takes a state.
yeah, unless the muley becomes incredibly like interesting to like the Army Corps of Engineers, I doubt we don't get any money to look at its roots.
Riverbanks, you can go down a train and you see riverbank cuts. Some of those roots go 15, 20 feet deep. So the kind of soil is called crocovina. It's sort of drying, swelling the soil, things go down, keep going down.
The grasses are down 15 feet. Do you have pictures of that?
Take it down here.
I would very much appreciate it. Where's this at?
Well, Green River.
Okay. Yeah, I just haven't seen any of this stuff, so this would be really good to see.
All riverbanks give you a good idea of the root cover, how deep they go. And even with trees, up on the limestone bluff where we live, We've got post oaks growing up there on limestone. They make their own soil as the roots grow.
Well, and I've seen from road cuts the kind of depth that the trees can get, but I haven't seen, like, most of the road cuts I have only have kind of a shallow root system, and it's also a road cut, so I can't get super close to look at the little stuff.
The water down through the limestone.
No, I would love to see that because my idea was that they were kind of doing it similar to the trees of going deep, but I haven't been able to prove it. So this is very good, especially because my boss doesn't think that they go deep. So this is actually very helpful.
We're all gonna be sending you pictures of grass.
Right. If you see any deep grass roots, send them to me so I can prove to my boss that they go deep. I got one more.
That slide you showed a few minutes ago about the different plant roots, there, looks like something I've seen before. Is that based on, it was, you saying that was based on some hypothesis of how they adapt to, low water conditions?
Yeah, I think it's that and just the diversity in form, like of rooting systems is kind of what that figure is supposed to illustrate. But that's not my figure. All right. Thank you very much.
Comment I'd like to make is, I've heard talks by a number of grad students, and so many of them so technical, and in all the lingo of that subject, that I came away not knowing what they were even talking about. And in your talk, I was able to follow everything.
Thank you. Good. I've been working on that.
People that aren't other grad students.
The last time I gave a talk like this, I was criticized for doing that. So I tried to not do that this time. So I'm glad it came off and like understandably.
Speaking to non-specialists is a specialty all its own.
Well, I would consider you all specialists over me. You guys know way more about this. I'm not from here, so I'm still learning about the plants here.
You don't have a chance to get down to around San Antonio area.
I went on one of the hikes at the symposium last fall. Was it San Antonio last fall? Was that? Or New Braunfels? That's the closest I've been.
There's a whole karst system down there north of, some government installation. Camp Bulls.
Okay.
Yeah. All water in